My name is Zheng from Department of Mechanical Engineering, University of Michigan. It is my great pleasure to give this presentation. Microengineered stem cell models for synthetic embryology. First, a very brief background introduction, just to make sure we're on the same page. The first few weeks of human development after conception are crucial for successful pregnancy and fetus health. Due to the scarcity of human embryo samples, the critical time from 7 days post-fertilization to about 28 days post-fertilization is often referred as the black box period. During this period, many important developmental processes happen. So far, most of our knowledge about early mammalian embryo development is obtained through studying mouse models. However, there is growing realization that significant morphological and genetic differences exist between the two species, which makes cross-species comparison quite problematic. This is where I will contribute. I build synthetic models using human stem cells and micro devices to open up a window to examine this previously inaccessible period of human development. I believe these stem cell models are the best demonstrations for the emerging field of synthetic human biology. Of course, my research so far has been focused on embryo development, but the potentials are certainly beyond this. I recently developed a synthetic embryonic sac model. The micro device is inspired by Professor Roger Kemp's angiogenesis devices. It contains 3 parallel channels, partitioned by supporting posts. The central channel is preloaded with gel, while the other 2 open channels are used for cell loading and chemical induction. Stem cells are allowed to settle into the gel pockets and form cell clusters. This setup allows us to stimulate just one part of the stem cell cluster by introducing morphogen to only one of the open channels. In this image, the top is from monkey embryos at day 13. This is the amnion and this is the epiblast. The immunostaining shows the markers of primordial germ cells. That's right, we start developing germ cells when we are still an embryo. The bottom is the synthetic model made with stem cells. We can see in terms of size, shape, and even the germ cell specification, they are very similar. In addition to a great resemblance with the real embryos, the reproducibility is also superior. The video here is showing synchronized development of embryo-like structures within the same microfluidic device. And not only is the morphology highly reproducible, but also the cell fate. As shown in this immunostaining image, green is for amnion, purple is for epiblast. And because of the reproducibility, if I want to make a lot of them, I can easily make a lot of them. The stem cell-based model is not another organ-on-a-chip device. Of course, the devices may look similar, but in terms of in vivo relevance and impacts on related biological research, my system is on a different level. For instance, it enables many mechanistic studies previously unimaginable due to the lack of human embryo samples. We can study the developmental trajectories and the genetic networks of certain developmental process. Specifically, we can conduct single-cell RNA sequencing at different time points, wherein individual cells in the devices are sequenced to obtain the genome-wide transcriptome signatures, and use machine learning and statistical algorithms to review the trajectories of certain cell types and the key genetic network governing this process. And if you are interested in certain genes function, we can just generate a genetically mutant stem cell using CRISPR technologies, which are getting very standard these days, and repeat the protocol. You will have a genetically mutant human embryo-like structures. Then it is very convenient to examine the morphological and the genetic phenotypes. In this case, nodal gene is knocked out. We can see the migration of gastrulating cells are significantly affected. We can also study the cellular or tissue crosstalk, which are too complex to investigate in the native tissues. Like in here, we observe that T-positive gastrulating cells always emerge from the junction of the amnion, which inspired us to look into the inductive effect of amnion cells. And then we for the first time proved that amnion functions as a signaling center in the initiation of human gastrulation. It is a brand new discovery to the developmental biology community. The synthetic model also has potentials for translational applications. It can be used as a drug or toxicity screening platform to examine the potential negative effects of certain medications or environmental factors on human embryo development. We have filed 2 patents and initiated the process. Here, let me make it clear. I am an engineer who is passionate about biological studies and strongly believe with the unique tools I have, I will be able to make more important biological discoveries even ahead of the top biologists. But the focus of my research is still technology or model development. Here I will introduce another example, synthetic neuron tube system. The mammalian central nervous system arises from a neuron tube. The patterning of the neuron tube is critical because it defines the specialization of different regions of the future brain and the spinal cord. In this schematic, it shows the early regionalization and the key markers of different regions of the neuron tube. The micro device I used here is a modification of the previous one. This time, the stem cells are injected into the central channel, and they will only adhere to the microprinted stripes and then form colonies. After I introduced the collagen gel into the central channel and added neural induction medium, the stem cell will differentiate into neural cells and, spontaneously form 3D tube-like structures. By applying morphogen gradient, we can obtain the same patterning with the real neuron tubes. This image shows the staining of several key regional markers. OTX2 marks the forebrain and the midbrain region. HOXB1 and HOXB4 label the hindbrain. HOXC9 marks the spinal cord. In the bottom, PAX3 and OLIG2 indicate the dorsal and ventral region of the spinal cord, which will further develop into neural crest and motor neurons. Of course, this synthetic neuron tube system requires further characterization, like single-cell RNA sequencing, but these staining images are very encouraging. Just as the synthetic embryonic sac, there are many potential applications, since, as being said earlier, the biological community almost knows nothing about the black box period. For instance, we can study the genetic and the environmental causes of the neuron tube defects of the newborn, and we can also study the connection between certain neurological disease, such as Huntington's disease and Parkinson's disease, with neuron tube early development. I'm also very interested in developing the synthetic placenta model using trophoblast stem cells and have achieved some preliminary success. We do not have time to talk about it. The related contents are included in my research statement. A very brief summary. I will combine approaches from microengineering, stem cell biology, and bioinformatics. The goals of my research are to establish stem cell-based synthetic models for developmental biology, stem cell biology, and disease modeling, and to apply these models to reveal the most fundamental mechanisms unique to human systems.